Hey, hey, hey! I'm so excited that we've made it to the final lecture in your Anatomy 201 class. As you watch this video and go through the PowerPoint slides, take out your notes and take additional notes on your on the Word document that you found on Blackboard. The video will just consist of pictures, some words, but mostly pictures, and you want to have those words out that are on the Word document so that you can follow along and know exactly what I'm talking about. But before we start, make sure that you have watched the video that's linked right here at the top. It's a Bozeman Science video, and I think you'll enjoy it, and it'll help you get a foundation before we even start. So if you haven't already, stop this video and watch that one before you watch this one. Okay, let's go ahead and get started. This chapter is all about our senses. And we know, I just thought that picture was cute, but we know what our five senses are since elementary school. We taste, we see, we can hear, we can smell, and then we can touch, right? So those would be the five special senses. And then we have lots of other ones, like you just learned in the video from Bozeman Science, like temperature, thermoreceptors, and pain receptors. And so the first thing on the paper says that sensory receptors are structures that are specialized to respond to change in their environment. So that's really the key of the whole chapter. Okay, these are five examples that we're going to talk about. The first one is a chemoreceptor, and you want to know, this is on there, so you want to know this, but that's a change in chemical. So you'll see chemical substance right there. So you need to be able to pick out a chemoreceptor is a change in chemical substance. Okay, so that would be the example of, it says chemoreceptors in internal organs can detect change in blood concentration of oxygen, but anything. So oxygen, blood glucose, sodium, potassium. So you have receptors in your body that help you maintain that homeostasis, right? Okay, thermoreceptors are going to be change in temperature. You want to pick out chemical, thermoreceptor, change in temperature. Photoreceptor, change in light. So make sure you know photo is change in light. And that's going to be two photoreceptors that humans have, are the rods and cones in the back of the eye, right? And remember from lab, cones help you see color. Rods allow you to see black and white. If you don't remember, we're going to get to it in this video. Okay, nociceptors, those are also called pain receptors, and they're going to be a response to, highlight for me, tissue damage. Nociceptors, also called pain receptors, so you want to know all of that, are tissue damage. And then mechanicoreceptors, that's going to be to touch. That's your response to touch. Okay, so if this were to be matching on the test, could you pick it out? Chemoreceptors are chemicals, thermoreceptors are temperature, photoreceptors are light, so respond to change in light, nociceptors are tissue damage, and mechanicoreceptors are touch. And that last one should just be touch highlighted. Okay, so pause the video, make sure you got all five types of sensory receptors and then what they respond to. Sensory receptors can be nerve fibers, and it can be peripheral nerves. It says peripheral nerves transmit sensory impulses to remember that stands for central nervous system, where they are analyzed and interpreted by the brain. And then, of course, you know what a sensation is. It says it's an awareness of change in the external or internal environment. And so it's sensation is where you're aware that something has changed, right? You feel a sensation coming on. Okay, these are the four senses we're going to talk about. We're not going to talk about touch. So there's taste, sight, hearing, and smelling. So the first one is taste. And so make sure you're looking at your paper. But a sense of taste, we all have these taste buds. So you can see right here in the picture the taste buds. right here. So that would be one taste bud. And those are sensory, so the sensory receptors for taste are down inside those taste buds. Located primarily on the surface of the tongue, the taste buds are. Taste buds are scattered throughout the roof of the mouth, the lining of the cheeks, and the walls of the pharynx. So the sensory receptors are on the tongue, but taste buds are actually all over the cheeks and walls of the pharynx. Taste buds are associated with elevated ridges called papillae, and that's what you can see in the picture there. You can see it's listed there. Okay, the four basic sensations that we're going to talk about. Taste sensations are sweet, are located at the tip of the tongue. So you probably have heard something like this before. So sweet is at the tip of the tongue. Sour is located on the sides of the tongue. Sour taste is usually associated with citrus. Oh, that's labeled number 3 on your paper, and it shouldn't be. Okay, salty receptors are located on the tip and the sides. And then bitter are located in the back. So if you were to go ahead and figure this out, you got sweet on the tip of the tongue, and I'll write that in for you. Okay, sour, sides of the tongue. Okay, salty is going to be the tip and the sides. And then bitter is going to be in the back. And that one's easy to remember because B and B, right? The bitter is in the back. So that's where those sensory receptors are actually located. So when we're thinking about tasting stuff, the taste buds are in different locations. Okay, with smell, you definitely want to know, and I left it the title of this slide, olfactory, that means smell. Right, so with smelling, we know much is unknown about exactly how we discriminate, like strawberry versus onion. There's a big difference, right? But researchers have discovered that it has to have a liquid. Well, wait a minute. Liquid, think about it. I can smell coffee beans, and they're not a liquid. I love coffee, right? Lots of things we can smell that are not liquid. And so how does that work? Think about it for a second. If we can only smell liquid forms, but we can smell things that aren't liquid, right? The liquid comes from inside your nasal cavity. So even though I smell like coffee beans that are not liquid, the mucus in my nasal cavity, and I'm kind of stopped up right now, you might can tell, it says the clear mucus dissolves the odor that are not in watery form. So that's kind of interesting. The nose contains specialized sensory nerve cells or neurons that are hair-like. They are cilia, right? Little bitty hairs, which you can see in the picture right there. Okay, and then that third bullet is pretty important. Each neuron, so highlight this one, each neuron sends a nerve fiber called an axon, we know all about axons by now, to the olfactory bulb. You got to know olfactory is smell. A brain structure just above the nose. So just above the nose, and you can see it in this picture here. We'll highlight it. Look right here, olfactory bulb is just above the nose. And that allows you to smell. That's the nerve fiber. So each neuron sends a nerve fiber, called an axon, to the olfactory bulb. So that is part of the brain, that olfactory bulb, that helps you smell. If the mucus in your nasal passage becomes too thick, then you can't smell. You know how it is when you're stopped up, you can't smell. Right? A lot of times when you're sick, you can't smell things. Your brain receives no signal to identify the odor. The olfactory sense peak around 20 years old. So I guess at 20 years old, you're smelling your best. You are able to smell the best. Not that your body smells the best. But as you get older, that decreases. Some people think that 1% of receptors are lost every year, but that's just an estimate. And then a substance called methylmercaptan is added to a natural gas so that it leaks from gas pipes will be noticed. And so oftentimes when we smell gas, and a lot of times here at Gas and State in the micro lab, there'll be, not a lot of times, but there has been before, a gas leak, and you can smell it because of that methylmercaptan. Okay, let's move on to sense of hearing. So that was all that, those words. And the only thing I've really highlighted on my paper is the olfactory bulb. So make sure that you've got that. There, so make sure you've got that one. Okay, moving on to sense of hearing now. So you should be pretty familiar with the ear from lab. You'll see the outer ear or the auricle, the external auditory meatus, right? So hopefully you're pretty familiar with all of this. The structures of the outer, middle, and inner ear are involved in hearing. So I'm looking at the paper now. The inner ear also contains structures to provide a sense of balance and equilibrium. Remember these semicircular canals, they have liquid in them, and that helps with balance. The auricle or pinna, that's this part right here, directs sound waves into the ear, so that's the outer part. The external auditory meatus, that's going to be this part right here, contains hairs and wax to help large foreign particles, like bacteria or bugs or flies or roaches. I know you've probably heard stories where bugs crawled into people's ears. Ooh. Transmit sounds to the tympanic membrane. Okay, so right here, now we're getting into the good stuff, is the tympanic membrane. So it's kind of like a little trampoline or eardrum, for lack of a better word, right? And the sound waves are going to vibrate that. So contains hairs and wax, but it transmits sound to the tympanic membrane. So when it vibrates, though, you'll see that it sends impulses to the malleus. So now look in red. That's the malleus, that bone. So look at the word. So I don't cover it up with marker. So you'll see the tympanic membrane, when it vibrates, it transmits sound waves to the malleus. So you want to know that, though. The tympanic membrane transmits sound waves to the malleus. That's the sixth bullet under hearing. Okay, the tympanic air cavity is filled with space, air-filled space in the temporal bone that separates the external from the internal ear. So we're separated here, which is good. And then there's 3 auditory oscles or bones. The word oscles, O-S-S-I-C-L-E-S, that's the 3 bones. Okay, and they are the malleus. So you want to know where these are for your lab exam. The malleus, the incus, and the stape. So we should have already looked at these in lab. There's your malleus. Then the incus. And then you'll see the stapes right there. So the malleus is shaped kind of like this. I'm going to draw them out for you for fun. The incus is almost like a hammer, if you'll remember from studying it. And then the stapes is like that. So make sure that you can identify all of those for the lab exam. And then also I'll give you a clue. This picture may be on the final exam. So you may need to study this diagram for the final exam. Make sure that you're familiar. Okay, so when you're studying this for your final exam, the picture, you want to pay really close attention to where the malleus is, be able to pick that out. Where the tympanic membrane is, you want to be able to pick that out. The incus, the stapes, and the semicircular canal. And then we haven't gotten into the cochlear yet, but you'll see how it's a snail. Can y'all see how it's like a shape like a snail? Right here, this part. So you should be able to pick that one out pretty easily. Okay, looking at the 3 bones. The malleus is attached to that tympanic membrane. The malleus vibrates the incus and the stapes. So it's like malleus, incus, stapes in that order. Malleus incus stapes. Ligaments hold the stapes to an opening in the wall. So the ligaments are holding them there. You know that ligaments hold the bones. That's old news, right? And then otitis media. That is a bacterial infection. So write that in for me. It's on the next page, but just for fun, write it in. Bacterial infection of the ear. Okay, and so I know you're looking at your paper, but that was otitis media. So O-T-I-T-I-S. And that's an ear infection, right? Bacterial infection of the ear. Okay. The auditory tube, which would be right here, this part right here. Can't see that pink too good on it, can you? Right here, that red. Connects the middle ear to the throat. So a lot of times your ears will be cracking and your sinuses will be draining. You can feel the mucus. But it allows air to pass between the tympanic membrane and the outside of the body. So there's no pressure buildup, thank goodness. It equalizes the pressure on both sides. Okay, when we look at the inner ear, you'll see that it consists of a labyrinth, and all that is just those semicircular canals with the lymph fluid in it. Remember, it's got perilymph, and that helps with equilibrium. Between the oscus and the membranous labyrinth is a fluid called perilymph with the cells in the walls of the bony canal. So it's just a fluid. And inside that is endolymph, and you can see all that in the picture. That's the dark purple endolymph. And then the labyrinth consists of, and this is all the parts. So you got your semicircular canal, so the whole thing is labyrinth. And then you got your semicircular canals, you know that from lab. There's 3 of them. Involved with balance. Vestibule, which is involved for balance, right here. This area. Cochlea, you want to know that for sure. Cochlea, snail-like structure, that is the, write this in or highlight it, organ of, oh, I don't have room. Okay, it is, let's try that again. It is the organ of hearing. That's your cochlea. So you want to know that. Snail-like structure, organ of hearing. Cochlea may be divided into 3 parts. The scala vestibuli, scala tympani, and cochlea duct. So you can see all that. Okay, and then the organ of Corti, there it goes. That's actually what, this is the cochlea rolled out, but that is what takes these vibrations from the eardrum and sends it to the nerves. So turns it into, converts the vibrations into a nerve impulse. So that's right where the nerve hits. So it's inside the cochlea. And looking at this picture, you can just see, this picture you won't see on the test or anything, but just so you'll kind of see it. There's the scala vestibule, and there's the scala tympani. So it you can kind of see it better on this picture. And that, is the organ of Corti, inside the cochlea, deep within the ear. Okay, things that you want to know for the ear, it says the clinical terms. So you want to look over all those clinical terms and know the definitions on the paper for your test. So tinnitus is ringing of the ear, so maybe after a concert or something. Vertigo, you probably heard of that, where you're dizzy. Okay, otitis media, we already wrote that in, so make sure you know that one. Bacterial infection. Otalgia is an earache, right? So highlight that when you want to know for sure. Earache, and it can be caused from an infection, but the infection is otitis media. The pain is otalgia. So know the difference in those two for sure. Otitis media is an infection. Otalgia is pain. Okay, eyeballs, onto the eye. Okay, you want to know this picture. You may see this picture again on the final. So take a look at it, slow down, go through all the parts. Okay, the sclera, that's the white outer layer of the eye. Highlight that. You want to know it. It's on the test. Let's see, I don't see it on the picture labeled, but it's this white part right on. Let me make that smaller. Maybe let's make it yellow. Okay. Right there, that all the way around. Oh, there it is, right there. Sclera. So that's the white outer part of the eye. The iris, that's the pigmented part right here. The retina, that is going to be this part right here that contains the rods and cones, the photoreceptors, right? Cones are color, rods are black and white. This innermost part right there. So I'm still in yellow. Okay, the lens, you know where that is, right here. Refracts light and focuses into the fovea centralis, so into the hole. It refracts the light and puts it into the hole. The pupil allows light to enter the eye, right here, so the opening. The rods, that's going to be black and white. Highlight rods, black and white, cones are for color. Rods are black and white, cones are for color, and I got a picture of them close right here. Whoops. So you can see the rods and the cones right there. And then, of course, you know the eyebrow. It's actually to get particles or sweat from falling into the eye. And then the eyelids, it's protection. We know those things, though. And then eyelashes, those are also to help keep sweat from falling into your eyes. Okay, conditions of the eye. So you hear far-sightedness and nearsightedness often. Far-sightedness, look at this picture. Maybe pause the video and look at it. This one, just looking at the picture. This is a normal eye. Oh, I lied. This is an eye that's elongated, so you can kind of see that it goes like a little bit longer that way. I emphasize that for sure. This was a normal eye in the middle, and then this eye is too short. Okay, so hyperopia, that's farsightedness. That's this one right here. Hyperopia. So too short, right? The eyeball is too short, and the image is focused at the point behind the retina. So that would be what we call farsightedness. Myopia, nearsightedness, the eyeball is elongated, so that's the first one. And you can see the pictures over here too. Okay, conjunctivitis, that's going to be what we call pink eye. Inflammation of the eye is red. Inflammation of the conjunctiva, that's outside the sclera. A stye, that is, it's like a big, it looks like a big pimple, but it's really sore. It's a bacterial infection of the eye, of the eyelid and an eyelash follicle. So it's like one of the little follicles is got bacteria down in it, and it causes a big knot. Okay, pink eye, that's a conjunctivitis, but that is by bacteria or virus. So conjunctivitis is just general due to allergies. And then pink eye is from bacteria. So those two are used kind of simultaneously back and forth, pink eye and conjunctivitis. Okay, glaucoma, blockage of drainage. So like blocking, I know a lot of times dogs get glaucoma. It's like a blockage in the drainage area so that the eyes are really dry. Bleeperitis is the margins of the eyes are inflamed. I'm seeing doubles, not due from alcohol though. Diplopia is double vision, so DI means two. You can remember that one for sure. And then the last one, you may have heard of this one before, retinoblastoma. It's inherited, so it's genetic. A highly malignant tumor arising from immature retinal cells. So tumors in the retinal cells. But you've probably heard of the word blastoma before, so it's just retinal blastoma. And that is all the clinical conditions with the eye that you need to know. All right, please let me know if there's anything I can do to help you. Don't hesitate to email me when you're studying for your final, and good luck. I hope you all do well.